So I'm Ed Levin, as you all know. I just wanted to preview the remaining couple of sessions we're going to have. On April 20th, Christina Meade is going to give a talk on her work with HIV and substance abuse comorbidity from social to societal networks. That'll be virtual on 20th from 2 to 3. And then on 27th, we're going to have a live talk by Rena Das-Eden from Penn State University. This is together with the Center for Study of Adolescent Risk and Resilience on parental substance abuse and developmental outcomes implication for prevention. And then that'll draw the curtain on the spring semester. The fall semester, we're going to have this combined seminar series together with all the other interest groups, and stay tuned for that. If you have any suggestions for people to bring in, each of the subgroups are going to have a nomination process for bringing people in. Please let me know. Okay, so today it's my privilege to introduce Dr. Leon Coleman, or I should say Dr. because he's an MD, PhD, and he earned these degrees at UNC and stayed at UNC, and is assistant professor of pharmacology, and he's also a faculty member of the Bowles Center for Alcohol Studies at our sister institution down the road. And today he's going to talk to us about immune contributions to alcohol-related pathology, ARBD and AD.
Cool, thank you. All right, thanks, Dr. Levin, for that introduction and for inviting me to share today. Like you said, I'm going to be talking about ARBD, which is alcohol-related brain damage, and AD, which is Alzheimer's disease. So two conditions that are related to heavy alcohol use. First, I want to thank my team. I'm going to thank God that I get to work with such wonderful people. It's a special time of the year, Passover Day, Easter on Sunday. So I'm real thankful that I'm blessed to work with such wonderful people, especially Ali Barnett, who did most of this work here. Imrai David and Lamar, also two students who've done work and collaborated a lot with Fulton Cruz over the years. Jian Zhao did a lot of our slice culture, and Leah Quinn did some studies too. And it's really just a great bunch of group of people having fun, and we are looking for postdocs. That's interesting. We have some funding from MD Serena as well. I have one disclosure to report on this work, a provisional patent application we filed with the university looking at targeting lysosomal lipid in Alzheimer's. So I kind of put things like this is kind of layers of the brain, right? And so I did for the trainees, just to show you never know. The question you always get is, okay, how do you know what project you'll take with you or will become your faculty position? But what I'm talking about today kind of started a bit in PhD years, and during residency and postdoc kind of moved into kind of neuroinflammation and neurodegeneration. And this kind of came together with Alzheimer's disease and alcohol studies together. I wasn't really planning to go here, but it's funny. That's the fun thing about science is you never know which way you're going to end up. But we also have some work looking at neuroimmune signaling effects of drinking and stress, but I'm just going to focus on these two projects here today. And really how neuroinflammation and neurodegeneration are linked in the setting of heavy alcohol use. And this is kind of what's called the cycle of addiction, right? You may be familiar with it, but it's thought people go through periods of binge intoxication, and then when they're not drinking, they go through withdrawal and they feel bad, they have negative affect, which promotes this craving phase and ultimately a loss of control. And so they go around the cycle over and over again. And so what we're hoping to do is find strategies to intervene at different places here. Because fundamentally, the brain has this inability to change once a person has alcohol use disorder. So we're hoping that if we can identify new, possibly neuroimmune targets, maybe we could intervene here and help people out so they can break out of the cycle. And so the first thing I'm talking about is alcohol-related brain damage. Okay, now this occurs in people that drink heavy alcohol or chronic heavy alcohol. And you can see here this MRI that there's loss of brain matter in multiple brain regions, particularly in regions that are associated with decision-making, behavioral flexibility like oral frontal cortex, memory, cognition like hippocampus. And I'm focusing kind of on these more frontal regions like OFC that are really important for being able to change your behavior. So this behavioral inflexibility is you learn to do something, but you have difficulty unlearning. And there's kind of two ways this is studied and how we look at it. One is reversal learning. So an animal learns a task, then you change the task, and how well can they change what they've already learned? And it's very similar to the extinction of a well-learned memory, where we use foot shot, for example, conditioned freezing, and how well can a rodent forget that strong memory? And we question, we're not really sure all the time how well drinking in rodents translates to humans. Do they drink for the same reasons? We're not sure. But some of these basic, neurobiological processes we think may translate. This alcohol-related brain damage, we see not just shrinkage on MRIs, but neuronal death in key brain regions. And this is human AUD over the frontal cortex, and this is tunnel stain with Leah Quinn. And we see that in alcohol use disorder brain, that there's an increase of actively dying neurons in neuronal death. And we also see this with, if we do cognitive fluorescence, it's kind of hard to see here, but many of these are neurons that are actually actively dying in the AUD brain. We want to have a model in mice that we can mimic this with. And here we do our 10-day alcohol model, binge alcohol model. Where mice are given 5 grams per kilogram per day, intragastric. And then at 24 hours, we harvest and look for pathology. These are in the binge range of alcohol peak BACs that are reached by humans. It's also important to note that mice metabolize alcohol 10 times faster than humans. They may have high peaks. Their average BAC during intoxication is around 0.1, which 0.08 is legal limit. So I think that's important to point out. And we do see cell death in key brain regions that we also see damage in the human brain. This is cleaved caspase 3 staining, in orbitofrontal, prefrontal cortex, interrontal cortical regions. We also see it with fluoro JFP as well. And if we do tunnel stain, we also see. So we do get good evidence of neuronal death in these key brain regions involved in behavioral plasticity and reversal learning, for example. So if we look at different behaviors, so this is reversal learning, and this is really linked with the orbital frontal cortex function as well as some other regions, but OFC is very important in this task. Basically, the mice get ethanol for a week, and then a week later, they do the Morse water maze and kind of learn to escape. And then week 2, we move the escape platform in the Morse water maze and see how well these mice can change their behavior. And this is just the learning phase here. And this is, control and ethanol. And you can see over 5 days, both groups really learn the task. There's really no difference in actual learning. But then when we switch the platform location, this is where differences arise. And you can see the white circles are ethanol. And the time to escape, it takes them more days to reach criteria. So there's a reversal learning deficit with chronic ethanol.
And where is that in the progression of the 10 days?
So this is a week after the 10 days they learn.
That's right there. Okay.
And then one more, sorry, I'll slow down. But then week 3 is when they reverse. So they haven't seen alcohol for 3 weeks at this point. So this is a chronic, a persistent effect. And this is just kind of a heat map showing the swimming zone, the controls. This is where the platform was when they learned it. And then after you reverse it, they learn, and they kind of, when they've learned it's reversed, they circle that location. But in the ethanol treated mice, they don't have that kind of strong preference. So a reversal learning deficit. What about the other way to look at, the other way we like to look at behavioral flexibility is with extinction of a well-learned memory. So we use conditioned foot shock here. So we do a memory extinction. So this was after they had their reversal learning test. So 4 weeks, I haven't seen alcohol for 4 weeks now. And it's basically a conditioned fear, conditioned foot shock freezing asset, where if the mice are in a context, they learn Pavlovian pairing of this tone with the foot shock. So they learn this association and they freeze. And with multiple pairings, you can see, the mice freeze. They learn to freeze. They learn that tone is predicting a foot shock, so they start freezing. So they're learning this association. But then after they've learned it, we put them into a different context and, just give them the cue. So they'll remember the cue will cause is associated with a foot shock. But we give them the cue over and over and over again. for them to extinguish it, so they can, it's like exposure therapy for PTSD or, something like that. Basically, you learn, okay, I'm not getting shocked anymore, so I freeze less and less. You can see this happens in the control mice, right? These are blocks of time batched together. But with more and more tones without shocks, they do start to freeze less. But the alcohol-treated mice, they don't really extinguish like that. So another index that once when they learn something strongly, the brain, they kind of get stuck there. And we think this is happening. You see these similar kind of deficits in alcohol use disorder. But the brain is not as flexible, and they've learned this behavioral pattern. It makes it extra difficult to now change your behavior. And
these are all young adults.
These mice, these are like 8 weeks when they start.
In the male or female?
We've done both males and females.
Yeah. Both. You didn't say sex difference?
Not in this behavior. No,
Oh, okay.
I haven't seen any sex difference.
8 weeks, that's like late animals?
Well, so we give them at 6. They have a week of habituation, and we start.
At 6 weeks?
We acquire them at 6 weeks.
Oh, you get them at 6 weeks.
So they habituate for a week. So at 7 weeks, they start their alcohol. So that's 10 days. And
That's
then,
45, that's late adolescence, yeah.
no. They're 60. They're over 60 days. When they start. They're adults. They're adults. I'm maybe doing my math wrong, but they're 60 days old when they start. These aren't.
When they start,
They
wasn't
start.
it the same in my sister? It's like 70 is early adulthood.
So
90
60,
is full adult.
60 is like early adulthood in mice.
Yeah.
Huh, okay, cool.
So then the question we start asking, okay, how is alcohol causing this pathology, this cell death, and this behavioral phenotype? So one thing that we find at the same time is we see cell death, we see a lot of signs of neuroinflammation. We see changes in microglial shape. This is, structural changes in microglial, consistent with microglial activation. We see induction of pro-inflammatory transcription factors like phosphorylated NF-kappa B. So we started to ask the question, we don't know what's chicken, what's egg, right? And so we started to want to answer the question, well, does this inflammation promote the neurodegeneration caused by alcohol? And so another feature of the neuroinflammation seen in alcohol use disorder is toll-like receptor signal. And so toll-like receptors are what's called pattern recognition receptors. So they're immune receptors. They recognize certain motifs, structural motifs associated, for example, with bacteria or viruses, RNAs, and to initiate immune responses. And in the human AUD brain, all of these have been found to be increased. So we wanted to see, okay, we know that toll-like receptors are induced, and some of these, like toll-like receptor 7, have been found in the setting of HIV to cause neuronal death. So we want to look at toll-like receptor 7 for that reason. And the interesting thing about toll-like receptor 7 is that there's an endogenous microRNA that can activate toll-like receptor 7. So toll-like receptor 7 senses single-stranded RNAs, and this short single-stranded RNA, when it's encapsulated in a vesicle, it looks like a virus. And so it can actually activate toll-like receptor 7. And it's been found increasing CSF in Alzheimer's disease, implicating peripheral pain, TLR7 signaling, and trip channel association. So we started looking at this. Does LET7 and TLR7 signaling contribute to alcohol-induced neurodegeneration? In fact, it does. So first, this is a postmortem human alcoholic brain. We did staining for TLR7, and we found with the more alcohol use people had across their lifetime, the more TLR7 was expressed in the brain. And so we went to our model here again, our 10-day model, and we just started to look at toll-like receptor 7 in association with cell death. Again, like I showed before, we see cell death in these key brain regions of this model. We also see at the same time increased toll-like receptor 7. And there was some dose responsible because 4 days of alcohol versus 10 days, you were in an intermediate level of TLR7. So it really does kind of fit with what we see in humans. The more alcohol history they have, the higher the toll-like receptor 7 expression is.
This is acute.
This is again our 10-day.
Yeah, well, it's 10 day, but you have 24 hours of washout.
We look at 24 hours after the last
Yeah.
alcohol. And
Or
so,
4 or 10 day, right?
and so this is 24 hours after 4 days, 24 hours after 10 days.
So like acute withdrawal.
That's the time, yeah, 24 hours into withdrawal.
Have you ever looked at in terms of, is this repeated intoxication with alcohol or repeated withdrawal from alcohol?
Yeah, it's a good, I mean, it kind of depends on how you look at it, you know, so it's during withdrawal that we see certain key immune signals like TNF, for example, microglia activation kind of peak around 16 hours into withdrawal that we see. I definitely think there's a component of this multiple withdrawals. And so we have studies going where we actually do cycles of alcohol. So we do 5 on, 2 off, 5 on, 2 off over and over again to get that multiple withdrawals. Because I think that is like a kindling.
Isn't there an excitotoxic cascade that goes on?
There is, yeah. And I think there's kind of like a kindling effect, kind of like you see with seizures, with the more withdrawals that you have, that exacerbates that signal. So. And so if we do chrominofluorescence, we see this TLR7. We look at Newin. It's both in neurons and in microglia. Mostly in neurons, it's induced. So we wanted to look for the TLR7 agonist, LET7B, and we see that is also increased after our 10-day alcohol treatment. And so we wanted to now test this mechanistically. Okay, does LET7 TLR7 signaling promote alcohol-induced cell death? And so we use a brain slice cultures. And so these include like hippocampal and neuronal brain regions. And we do a 4-day ethanol treatment in the slice culture. And we really find that this really reproduces a lot of things we find in human alcohol use disorder brain and in our 10-day model. And then we look at withdrawal when you see the cell death. And so you can see this is control, and this is after ethanol treatment with withdrawal. You can see this is PI staining, and there's cell death in the neuronal cortex and hippocampal brain regions. And this is it quantified here. So what we want to do is a series of experiments, giving, blocking LET7, the TLR7 ligand, knocking down TLR7, and giving a small molecule antagonist to TLR7 to see if those would have any impact on the alcohol-induced cell death. And every one of them blocked alcohol-induced cell death. So if we block LET7, we block the ethanol-induced cell death. We block TLR7, we block it. And if we give this small molecule antagonist, we prevent the alcohol-induced cell death. So this was making us think this was going on, but now we're like, okay, what about in vivo? Okay, and this was M5049. This was developed by MD Serena. It's selective for TLR7 and also TLR8. It's an inhibitor. They're trying to. It's now in trials for COVID. They tried it with COVID. It was really developed for lupus, but they kind of contacted us because they wanted to see if it had any effects in the brain. So we were interested, and so we said, well, let's see if it can actually prevent alcohol-induced brain damage. So we did our 10-day model here with or without their compound, and just did cell death. And it's hard to see. Sorry, this is for J, but this green staining here. This is in neuronal cortex, I believe. And we see again with ethanol, we see our cell death. But if we give the TLR7 antagonist, we completely block that. And it's with Flora-J and with compound 2. So we think we have this, what we've identified as this pathway of ethanol is inducing LET7 expression in our secretion that then hits toll-like receptor 7, leading to cell death. And so we want to know, okay, where is this LET7 coming from? We know it has to be kind of encapsulated in the vesicle for it to work. Otherwise, it just gets degraded. So we did different cell culture lines. This is a neuronal-like culture, EV2 microglia-like culture, an astrocyte-like culture, and treated with ethanol, just see which would produce LET7. So we looked in the media, and there was no ethanol effect on neuronal secretion of LET7, but microglia really had a strong release of LET7, and a little bit from astrocytes as well. So this caused us to think that, okay, maybe microglia are driving neuronal death through the release of blood-7B. So in order to test this, we wanted to develop a specific way of inhibiting microglia. So we could give minocycline or other compounds, but those aren't necessarily specific from microglia. So we took advantage of chemogenetics here, where these are mostly used to inhibit neurons, which we all know, developed at UNC. But this is basically an engineered G protein coupled receptor, HM4DI. You can get activating or inhibitory ones. We wanted an inhibitory G protein coupled receptor here, and we want to express it only in microglia. So we used the CX3CR1 CRE2 line, which has a tamoxifen-inducible CRE and monocyte-derived cells, which includes microglia and peripheral monocytes. And we crossed it with the dread mouse, inhibitory dread mouse, which has a stop codon that's flocks prior to the dread. So when these come together and you get tamoxifen, you'll get the dread expressed in CX3CR1 expressing cells. And it has an M-citrine tag. So we did this in a slice culture first and added a 4-OH tamoxifen. And you can see that at rest, at baseline, there's no M-citrine, no dread expression, but after we get tamoxifen, we do get expression of the dread. And we see the tag. We did co-immunofluorescence for IgA1, and pretty much all of this was expressed in microglia. So now we have a tool here where we can give the ligand for this dread, which would inhibit internal calcium mobilization in microglia, which is required for them to change their effector functions, and see if we can prevent alcohol-induced inflammation and cell death. And so first we want to check and look at alcohol-induced induction of pro-inflammatory cytokines. And so you can see ethanol here is inducing TNF-alpha, MCP1, but when we add the CNO, which is the ligand for the inhibitory drug, we can abolish that. So we can successfully block alcohol-induced pro-inflammatory activation in this mouse. So what we wanted to see, okay, would this have any effect on alcohol-induced cell death? So we're back to our slice culture and treated with ethanol, did our 4-day 24-hour withdrawal. Again, this is a low carbon. You can see PIs for granulinitis, and you can see cell death here. But when we add CNO, we can pretty much abolish that. So by inhibiting pro-inflammatory microglia activation, we can protect against alcohol-induced cell death. This is a little surprising, because we thought it was just, for a long time, the Lord's been just excitotoxicity. But excitotoxicity does occur here, but this really implicates microglia in that process.
So do you see a similar effect in terms of protecting against cell death if you just give a GABA agonist or something like that?
We did not do that. I'd have to look back and see all the things that we did give. I don't think it protected against glutamate. I have to double check. We did a few things, but we didn't do all the things.
I have a question. So is this just like your example slides? It almost looks like there's nothing there. How do you find your regions? It's like easier to see on the scope, or like
Yeah, we can,
you
can see the slides. Yeah.
You can still see the slides. Okay.
Yeah, but this is the
Like, how do
actual.
you find out where you're at?
Yeah, this is the staining. This is the fluorescent staining. It's made
Oh,
black
it's
and white here.
fluorescent staining, and then
Yeah,
you change
yeah,
it
it's
to
the black and white. Yeah, yeah.
Okay. I was thinking it was like dab or something. I was like, how do you even find it?
Yeah,
okay,
it's
this makes
dye.
way more sense.
Yeah,
Yeah, right.
it's all
It's
that dye.
Agree. Okay, yep, fluorescent. Gotcha.
Yeah, sorry about that. So then we want to go, okay, what about in vivo? So we want to do the same experiment in mice with our 10-day model. So we took mice with the dread, gave them tamoxifen for 5 days. And now since this will also, cause CRE expression in monocytes, we wanted to wait 4 weeks to allow those to turn over. And so therefore, pretty much the only cells remaining that have your dread expression will be microglia. Maybe some resident macrophages, pretty much mostly microglia. And then so we did our 10-day ethanol with CNO, and we gave our CNO 10 hours after ethanol, kind of that point that he raised, because it's during the withdrawal that we see TNF and things going up. So this just confirms, we looked at pro-inflammatory cytokines, TNF-IL-1 trail, which is another one that we like, and the CNO blocks the induction of those cytokines. And we also looked at microglia activation. It's hard to see here, sorry, but this is kind of shape change of microglia. So you can see control. You probably can see from there that the processes are more pronounced with ethanol, but when we gave CNO, it blocked that. So you can see here. So we think we are blocking the pro-inflammatory activation. Now, what about the cell death? We then did tunnel. This is the orbitofrontal cortex. And really, CNO not doing much at all. When we give ethanol, we see a lot of tunnel staining, a lot of cell death going on in the orbitofrontal cortex that was prevented by giving CNO. So by inhibiting microglia, we're able to prevent alcohol use. So what does this happen? Does this relate to the behaviors at all, right? So that's orbitofrontal cortex. Does it relate to cognitive flexibility at all? So we did our same experiment, but we took them through those same behaviors that I showed earlier. And there were a couple that really jumped out. One is an anxiety-like behavior, so thigmotaxis, center time in the open field. Ethanol really reduced, and again, these guys haven't had alcohol for weeks. But the mice are still have more anxiety behavior. Ethanol reduces time center in the open field. But if microglia were inhibited during alcohol, this was prevented. And as far as behavioral flexibility, again, we did our conditioned fear memory extinction, and our control mice were showing some extinction after they learned the fear memory. Ethanol, really nothing at all. But if they had ethanol with CNO, they would extinguish nicely, a little bit nicer than control. Really implicating the microglial activation in this cognitive behavioral and flexibility associated with long-term alcohol.
Wait, sorry, when were the behavioral tests done? And are they the same mice in both?
Yeah, these are the same mice. So this is the same paradigm as before I showed earlier.
Yeah.
So they have the treatment, and they have, it's like 4 weeks of behavior after it. So this was done 4 weeks after alcohol.
4 weeks
Month
after.
after.
Wow, that's not.
So this was done like 2 or 3 weeks after. Yeah.
And was the CNO given every time you gave them alcohol?
With every alcohol, yeah, 10 hours after every alcohol, yeah. So,
Students.
Preventative.
yeah.
Yeah.
Yeah, so maybe anti-inflammatories during your hangover period could be productive, so.
Did you ever CNO group by itself?
In this one, we ran out of mice, so I had to leave, with that one, unfortunately. For the cell death, we had CNO alone. And
Yeah, because for something that just has such beautiful effects, then it raises questions of, where's the rub?
Yeah, what's the downside?
Yeah, exactly.
Was that the regular CNO? So it
Yeah.
has to go in DMFO and stuff?
And this is water soluble.
Water soluble. Oh,
Really, that's easier to use.
yeah, it's way easier.
Yeah.
And I think that one, we're just looking into it now, so I just have this on my mind, but it creates less clozapine. The water soluble.
I didn't know that. I didn't know.
Breaks down. I think that's what I found is that it makes less clozapine. So you're less likely to have this maybe off-target
Off-target
effects of
effect.
your, so this could be like a
That's interesting. Yeah, I didn't know that. Okay, so just kind of wrap up the first part of the talk was basically alcohol is causing pro-inflammatory microglial activation. Microglia secrete a microRNA within an extracellular vesicle, Let7b, that can activate TLR7 in neurons. This can lead to neuronal death, probably dysfunction as well. We haven't fully looked at all the effects of neuronal firing, but that's something we're in the process of doing. And behavioral inflexibility, which is a feature of alcohol use. So, when you think about neurodegeneration, you think about cognitive dysfunction, you kind of have to think about cognitive decline, things like dementia. And that's kind of where I'm headed in next with this. Because, there's a strong association between heavy alcohol use and dementia. And it's becoming appreciated more and more now. There's this study here, but a lot of epidemiology supports the association. But this was a study done in France with over a million hospital patients, and looking for the they thought, because they have great records, they could see who got diagnosed with dementia, and they could link their modifiable risk factors to dementia risk. And so we all know things like smoking, inactivity, obesity, these are modifiable risk factors for dementia and Alzheimer's disease. But what popped out in their assessment was that heavy alcohol use was the #1 modifiable risk factor for dementia and Alzheimer's, particularly early onset, okay? So this is a pyramid graph, and these are individuals, this is everyone who ends up with a diagnosis of dementia kind of across their age as they get older. And the red are people that had alcohol-related brain damage, and the yellow are people who had other alcohol use disorders or alcohol misuse. And you can see a really big portion of 69%. So, a really big portion of people that had early onset dementia. Yes, sir.
What do you mean by heavy drinking? Is it like more than 4 drinks a day or binge drinking? What's the definition?
Yeah, so here they were looking primarily for diagnoses of an alcohol use disorder. So, which now, is very easy to get, but if I'm remembering correctly, I'm thinking, I have to double check, but for some reason I think it was like 7 drinks or something on average a day, I think. I have to double check that.
I mean, that makes sense for France, right?
Can't remember. Yeah, I have to double check the number. Everyone wants to know the number. We all want to know what our risk is. It should be a risk
It's
calculator.
France, like a couple bottles.
But,
Russia.
Yeah, and Germany.
yeah. But so this was kind of impressive to me. Yeah. Another one was the Hunt study. This was in Sweden, I think, or Sweden, where they looked at earlier alcohol use and they found that midlife drinking. And this was more than 4 drinks a day. I remember that one. Increased Alzheimer's risk. I mean, so this was going back even like 25 years prior to diagnosis. So there's something, that's where a lot of these Alzheimer's risks, it's obesity during midlife. Obesity when you're older is not a risk factor, but obesity during midlife is. There's something about midlife that really determines people's trajectory as far as aging. High intensity exercise is really only beneficial during midlife.
I cannot quit playing basketball.
Can't go playing basketball.
Takeaway.
So midlife it depends on how old you are, what midlife is, right? I think really midlife is your late 30s to 60s, so like 40s to 60s. That's really midlife. So.
You're okay.
Just for a little while, though.
I mean, I have to wait. These guys are saying, you're not there yet. I gotta work.
You guys are locked
You're locked in.
in. Leon's like, calm down, class.
It's good. Oh, this is fun. So we were like, okay, what's the connection, right? And so we were wondering, well, maybe it's neuroinflammation, right? Because a lot of the things that we find in alcohol use disorder, alcoholic brain damage, they're also been implicated or, they're increased or altered in Alzheimer's disease as well. So, and this is not meant to read it, but this was, a table that we made looking at all the similarities between alcohol use disorder and Alzheimer's disease, and there are a lot, as far as neuroinflammation goes.
So is alcohol-related brain damage during aging similar in terms of the amyloid plaques? And
So I'll show a little bit of data on that. Yeah, so people with alcohol use disorder, they typically don't have full amyloid plaques. They don't have Alzheimer's disease. But I think it's a risk, just like hypertension, hyperlipidemia. These are risk factors for vascular disease, cardiovascular disease. I think this is it's a risk environment within the brain. But I'll show a little data on that. So, like I said, binge drinking early in life or in adulthood even. And so this one to show, this is kind of an update a few years ago on the progression of Alzheimer's disease. So there's this preclinical period, which is midlife, basically. And then people start having symptoms, non-cognitive impairment, and then symptomatic out here. But a lot of the damage is occurring in this period here. And, for example, amyloid starts coming up during this period and it's accumulating within neurons. Before you ever get a whiff of a plaque, amyloid accumulates in neurons. And that's the problem, I think. We have to block that process. inflammatory activation happens at this time too. So this is the period where we're looking at the alcohol effects, and this is where we think we want to intervene as well. And so here you start seeing intraneuronal amyloid, not plaques, but just intraneuronal, and early tau species. So there's a whole progression of tau phosphorylation. There's early tau phosphorylation species during this time. And so just to answer your question, this is alcohol use disorder brain, and there are increased kind of early AD markers in the human alcohol use disorder brain. So we found out that these phosphorylated tau species, these early phosphorylated tau species, are increased in the human AD brain, cortex and hippocampus, and A beta 142, which is a toxic form of amyloid, is also increased in alcohol use disorder brain. So, you know, this environment was there. But something else has to go wrong. Okay, so we started saying, what are the effects of binge ethanol on this early Alzheimer's pathology? So we kind of start out with, we did two models. We used this triple transgenic 80 mouse model, which, you know, this has mutations in amyloid processing and tau. Mice don't get Alzheimer's unless there's some sort of mutation. And the way we're really using this as a model of the clearance of amyloid and tau. This is how we're using it. Okay, so not an etiological model or pathogenesis model, but, you know, we're looking at how these things are cleared in these mice. So we either have adolescent or adult binge exposures that we've done, kind of following the epidemiology. And we're focusing on early Alzheimer's pathology, right? Intraneuronal amyloid. And again, this is just a mouse model, and this is just to show that we're looking at ages prior to the development of severe pathology in all of our tests. Like that. And we're seeing if alcohol can accelerate this progression. And so in our adolescent exposure, so this is the Nadia, well, this was, you know, essentially became the Nadia protocol from back in the day when we started these studies. But this is the treatment we do. 2 days on, 2 days off during adolescence. Then we stop and wait, you know, till P200, so they don't have alcohol for months. Then we look at pathology. And so when we look at P200, in this model, primarily females have much worse pathology, and that's where we saw the deficits after adolescent alcohol. And we can see increased amyloid A beta and tau species after adolescent alcohol. This is the P200 again. We look at specific brain regions, we can see this is intraneuronal amyloid. So this is subiculum, and you can see that the amyloid is not plaques, but this is within neurons. And it's increased after ethanol. And we also saw that in the size is so small, also that in the amygdala as well. So multiple brain regions, adolescent alcohol followed by a long break, you've accelerated, increased. Now what's going on? I mean, this is a transgenic model. Gene expression hasn't changed. Why is there so much more amyloid? So we started looking, again, is neuroinflammation involved? So we saw several pro-inflammatory cytokines that were increased persistently. This is hippocampus. And what we saw is that the expression levels of these pro-inflammatory cytokines were strongly correlated with amyloid and tau levels. It's making us think maybe there's an association. Again, is it chicken or egg? So we did another experiment that if we block pro-inflammatory signaling, can we prevent the persistent, this increase in AD pathology? And so here we did our treatment again, but we gave minocycline this time to inhibit microglia. And we saw that it completely prevented this increase in amyloid and tau pathology. So this is an amygdala here. After our adolescent intermittent ethanol, you can see a whole lot of neurons with amyloid inside them, but that's prevented by minocycline. There were also improvements in memory, so memory retention was improved, so this is also functional. So we wanted to ask in the last part of the talk, okay, how does this happen? Okay. And we also want to see if this occurred in adults. So we did our adult exposure. This is from 9 to 11 months of age. This is 5 days on, 2 days off, 5 days on, 2 days off. To give, you know, the mouse a break on the weekend.
The mouse.
Yeah.
Experiment.
But also this is cycles, this repeated cycle.
Work-life balance.
We also want to keep the cycling part of it. And we saw, increased amyloid, like with about, multiple brain regions. So again, it's intraneuronal amyloid primarily. There were some plaques at this age, but really not a whole lot of plaques at this age. The amyloid was increased in rhinal, frontal cortex. And again, no differences in gene expression. So it's not because the transgenes are being expressed more. There's something wrong with what we presume to be the clearance of amyloid. So we started looking at clearance mechanisms. And amyloid is a cell membrane protein. And it goes through cycling, like a lot of into the endosomal system, and a lot of proteins are. But when it's accumulated, these endosomes fuse with lysosomes to degrade amyloid in normal function. So we wanted to see, okay, is alcohol disrupting this, autophagosomal or lysosomal flux? And it is. So P62 is degraded by lysosomes. So when P62 is increased, there's an issue of cycling through the lysosomal system. P62 is increased. Becklin, which is an initiator of this process, was down. LC3A was accumulating. WAMP1, which is the lysosomal marker, is accumulating. The way to interpret this, there's basically a decision tree to interpret this. This is a complicated system. But basically what it boils down to is this pattern configuration points to lysosomal impairment. So you get P62 accumulating, you get a backing up of the whole system, LC3 accumulates, and then you have a compensatory downregulation in Becklin. So this is a kind of a common kind of signature of when you have lysosomal dysfunction. So we wanted to start probing lysosomal function with ethanol more. We looked in our slice culture. Sorry, this is so small, but this is still in the cortex, sorry. Several of the ATPase genes that acidify lysosomes, their expression was down. And I can't see this, but I promise you this is in the slice culture. This is lysotracker staying for lysosomal acidification. It was reduced by ethanol. So there's definitely some sort of lysosomal aberration going on here. So we kind of went on a hunt, right? We're like, okay, why are lysosomes not working? So we started looking at a whole bunch of genes, right? So we looked at lysosomal fusion, autophagosomal fusion, like structural genes. And I have some data here from obese because in obese mice, we see similar enhancement in pathology. So we were trying to use multiple models to try and see if there's convergent cellular dysfunction that could be valid beyond alcohol in the population through multiple etiologies of Alzheimer's. And really, the one place where we saw like a commonality was this downregulation of LAL. Now, LAL is lysosomal acid lipase. It's a lipase within the lysosome. It's pretty much the lipase within the lysosome. This is interesting. And so we wanted to kind of wonder, is there some lipid dysfunction? There's some sort of lipid problem going on. And so what we see is that ethanol is actually causing what we call lysosomal neurosteatosis. So, I'm sorry, this is hard to see, but we were doing lipid stains in the brain after our alcohol treatment. And maybe you can see it here, but this is a lipid stain here. There's increased lipid in the brain after ethanol, kind of like what you see in the liver. And this was accumulating within lysosomes. So it's kind of hard to see here, but the white here is lipid within lysosomes inside amyloid-containing neurons. And there was an accumulation of lipid within lysosomes after chronic alcohol. That's kind of what this is showing. So this is brain.
Oh, I thought so.
Similar to,
I must have heard lipid and thought I heard lipid.
well, I said similar to because the liver, you get fatty liver. Right?
Okay, got you.
But you're not getting fatty liver, you're getting fatty brain too,
Okay.
apparently. So, yeah, so lipid within lysosomes here in amyloid neurons is increased with ethanol across regions. And if you look at where amyloid is, the localization within the cells, within the lysosome or in the extralysosomal cytosol, it was mostly in the cytoplasm. And when you give ethanol, there's less in the lysosomes. So you have lipid accumulating in the lysosome, but amyloid is not getting into the lysosome. So we started wondering if this accumulation of lipid was preventing amyloid from getting into degrading the lysosome. And we saw these strong correlations between lysosomal lipid and amyloid levels. The more lysosomal lipids you have, the more amyloid you have. The more lysosomal lipids you have, the more amyloid you have across brain regions. So, you know, what about microglia? Are they involved in this at all? So we went back to our slice culture. We treat with ethanol, and we see in the slice culture as well, there's accumulation of lipid. But if we inhibited our microglia, that was protected. So somehow microglia are promoting this lipid accumulation in neurons that's caused by alcohol. And so, okay, why is this happening? Okay, why? This is just to show we looked at a whole bunch of stuff across multiple models. So we looked at, you know, in our ethanol, high-fat diet, aging, our wild types with ethanol, which all show this increase of lipid in the lysosome. We thought, okay, if there's some underlying process to explain this, it would match across all these models, but nothing really stood out except for LAL. Nothing stood out. ER stress didn't stand out, differences in metabolism, beta oxidation, neutral lipases, nothing stood out across our models. There was some difference in some lipid droplet proteins, but nothing really stood out except for the LAL, the lysosomal acid lipase, was the only thing that was consistent across all our models. So it doesn't mean there's not something else, it just means everything we could look at. So we wanted to test directly, okay, if you lose LAL, does that have any effect on amyloid pathology? So again, with our ethanol treatment, this is protein now, we do see a reduction of LAL. We see the same thing in obese mice. If we look in human alcohol use disorder brain, we also see a loss of LAL. So about a 50% reduction in LAL in the human alcohol use disorder brain. So this happens in humans and in our mouse model. When we looked at, we wanted to see now, okay, is there an association between LAL and amyloid in the actual tissue? So we did co-staining across multiple brain regions in all our animals for LAL in red and amyloid in green. And you can see there's some plaques here at high mag. Sorry, that should actually have the scale bar. But what you see is in regions that have, you can't really, it's hard to see here, but where there's more red, there's less green, basically. Where there's more LAL, there's less amyloid. So there's more LAL here, less amyloid. Intermediate amount of amyloid, LAL here, intermediate amount of amyloid. A lot of amyloid, no LAL. So it really follows this inverse relationship, you know, looking at the cells even of more LAL, less amyloid, less LAL, more amyloid. So we said, okay, let's test this directly in our slice culture, okay? And so we made slice culture out of the Alzheimer's mouse. There is an inhibitor to LAL. And so we said, okay, let's give this, it's called LAL stat. First, let's look and see if lipid accumulates. It does, as expected. In the dose, the concentration of beta-marin, the more inhibitor of LAL, the more lipid. Okay, now what about amyloid? And that's where we saw that if the more LAL inhibitor inhibition, if you inhibit LAL, you get amyloid go up. So this is pretty clear here that, okay, and LAL does not degrade amyloid. It degrades lipids. So this is consistent with that hypothesis. And so we said, okay, we can go one direction. If we block it, we increase amyloid. If we supplement LAL, can we prevent its accumulation? And so there's a recombinant LAL that's available because there's a rare disorder, actually, people that have no LAL genetically. They die very young. It's a terrible disease. But they're trying to give people recombinant LAL to treat this. And so we went to our ethanol. And ethanol, again, increases lipid. If we give recombinant LAL, we can block that increase of lipid caused by alcohol. Now, what about amyloid? Ethanol, I don't know if y'all can see that at all, but there's increased amyloid here. There's green staining here that is totally blocked by giving recombinant LAL. So this lipid accumulation is preventing the degradation of amyloid. And then we finally looked into Alzheimer's brain. So we got a bunch of Alzheimer's disease brain and looked in several different regions. This is LAL staining. And you can see like neuronal shape here in the subiculum, in multiple brain regions. But in Alzheimer's, it's really almost all gone, and the neurons don't have that, there's no shape of neurons anymore. We think it's mostly in glia at this point. So there's a loss of LAL in Alzheimer's disease. Also in healthy subjects, it goes down with age. So in humans, and we did in mice, looked in aged mice versus younger mice, LAL is going down in neurons, lipids accumulating in neurons. So I think it's really a fundamental age-related risk factor for Alzheimer's disease. So kind of what I just kind of showed is that with ethanol and our obesity, we did some obese mice too. We get pro-inflammatory microglia activation. There's somehow it's disrupting neurons, leading to a loss of LAL and accumulation of lipid. Now this lipid accumulates within lysosomes, this loss of LAL, and somehow is preventing lysosomal degradation of amyloid. You get accumulation of amyloid and increase in early amyloid pathology. So just to summarize that, of course, I think, ethanol promotes neurodegeneration and neurosteatosis through pro-inflammatory microglia oxidation. This lysosomal neurosteatosis impairs the lysosomal clearance of amyloid. Loss of LAL promotes lysosomal neurosteatosis subsequently. And this LAL loss occurs with aging and may be a fundamental underlying risk factor for Alzheimer's disease. LAL neurosteatosis, I think, could serve as a new diagnostic marker and a preventative target for Alzheimer's disease and related dementias, and that's kind of what we're working on here now. And this kind of just summarizes that. Amyloid microglia are really important. They could be therapeutic targets for both alcohol use disorder and the alcohol-related brain damage and or Alzheimer's disease through blocking this microneuronal, like immunometabolic crosstalk. And that's all she wrote. So that's any question.
So what about the LAL? Is the ethanol having a direct effect on that, or is that just part of the cascade of, what's the kind of order of operation of ethanol?
Yeah, I'm not, I think it goes, my hypothesis is pro-inflammatory microglia somehow communicate with neurons to cause a loss of LAL in neurons. So, the fact that we also see it with just aging, with no alcohol, and I put that in here, but we just wild-type aging in neurons, LAL goes down with age, and lipid accumulates within neurons with age. And there's with aging, there's just simmering increase in inflammation. I think that may be involved. That's kind of the current hypothesis, working hypothesis.
Is this LAL mechanism, is this the kind of general, cellular stress? It's kind of like FOS or something. You're going to see it in reaction to a lot of things. Like if you have physical damage, or if you have illness, or if you have other sort of drugs. So is it going to be invoked as well?
That's it. I don't know. That's a good question. I haven't seen beyond the LAL insufficiency disorder. I haven't seen anyone look at that before, so I can't say for sure. But that's something we should, because if that were the case, then a lot of different exposures, like you're saying, injuries or depression or whatever could promote that.
Did you say one of the drugs you're using, the person who developed it was using it for other things, and they were curious as to whether... So
recombinant LAL, yeah, that was kind of generated for the LAL insufficiency disorder.
I see.
And it's kind of shocking to me that you can give this peripherally and it finds its way into lysosomes.
Yeah, that's a pretty big...
To me, that's hard to believe.
Yeah.
You know what I'm saying? It's kind of hard to believe. I was even shocked that it worked in the slice cultures.
Yeah.
You know what I'm saying? I was like, I kind of was, but it does, and the inhibitor does the same thing. It does it. And so it kind of,
They should just add it to whiskey.
yeah.
Yeah.
It might be expensive.
I bet.
But
Any other questions?
Great talk.
Great talk.
Really elegant, well-connected story.
Yeah.
Full circle.
Yeah, well, we'll see. I mean, hopefully, hopefully it'll work. I mean, we're trying to develop pet ligands, things like that. See if it can be used in people, but,
So are drug companies on the track here to have small molecules to affect the system?
well, we just filed our patent like 2.5 weeks ago. So,
I think
that's.
Ryan Roth should be involved with this, right?
Yeah, that's where we're gonna try and do drug screens and all kind of stuff to see if we can induce it. This kind of the next year we have to kind of try and get some of that done.
That's great work. Okay, thanks for coming.
Yeah, thanks for having me. Yeah, that's really